I think should wait for 2 more minutes before we start.
I think you can go ahead and get started.
Right. So good afternoon, everyone. My name is Sai. I'm a first-year PhD student in UConn Health. And I joined Beyond the PhD because I've always been interested in exploring careers outside of academia. And so I'm very excited to present our speaker for today, Dr. Emaly Piecuch, who, after completing her BS from Purdue University and her PhD from UConn Health in 2019, is now working as a genomics educator up at Maine at the Jackson Laboratory. So just some housekeeping rules before we start. So if you could please use the chat box to ask your questions for the speaker during the talk, and to please remain muted during the talk, and any questions can be asked after the talk is done. And we have some upcoming events before we begin. So if you're interested in exploring careers outside of academia, as well as one that's academic. So we have a talk by a research geneticist next month, as well as an assistant professor up at UConn Health. So if you're interested in joining, you could register for these events. We also have the summer office hours. So if you're interested in applying for a job and you want someone to review your job application or your resume, you could reach out to us, and one of the members of our group would work with you in a peer review session for around 30 minutes. This would be available until the end of August. And if you're interested in donating to our GoFundMe, we are hoping to expand and become a nonprofit. So if you're interested, you could please support us by donating to our GoFundMe. So on to you, Dr. Piecuch. Sorry, before we also go, if you're interested in joining us as a campus ambassador, or you want to become a career discovery speaker, you could contact us in our emails here, as well as our website. Right, on to you.
Awesome. Thank you so much, Sai, for that nice introduction. And thank you to everybody in the Beyond PhD program, Brittany and Rob, for the invitation. Just one moment and I'll share my slides and then get started giving you guys some details about my role here. Okay, how does that look?
That looks great.
Awesome. Thank you, Sai. All right. And also, obviously, thank you so much to everybody in attendance today. Looking forward to chatting with you. I have a couple of slides to show. I'm going to explain my background a little bit, go through some of the details of what I'm working on now. And then we'll have some time to discuss at the end any questions or questions you have for me or about the role. Awesome. So first, I'm going to give you my background in picture format, and then explain to you my current role and some of the things that I'm doing as a genomics educator right now at the Jackson Lab. And then I have some tips and tricks for you to find cool jobs at the end. Okay, so let's get started with my background. So I think I have 10 pictures here. So that's like 1,000 words a picture, so 10,000 words. I'll probably only say like 300 or less. So in high school, I was not really interested in science. I was interested in helping people. There's a picture of me working at the library. I worked in the picture department or the children's department, and I shelved picture books mostly. But here I am doing an outreach activity. So we were dressing up and reading stories to kids. So I really like doing that type of organizational community outreach stuff since I was little. And then on the right, you can see me there playing Glinda the Good Witch. So I was really, I guess, always interested in using my powers for good. So when high school finished and I realized I needed to go to college, it was a challenge for me to decide what to do, but I ended up choosing biology, I think, because I was just interested in the topics and helping people. So I guess I should mention that I was the first student or family member to go to college. So there was quite a bit of pressure to succeed, but I had some really, really great mentors and fun along the way. So as Sai mentioned, I went to the Purdue University in Indiana for my undergrad. And when I was there, I got involved in lots of community outreach. There's a picture of me showing some horseshoe crabs to some students at sort of like an open day. Here's me pouring some plates. So I got involved in molecular biology pretty quickly. I was very interested in that from the first course that I had taken. So I was lucky enough to get involved in pouring a bunch of plates there, as you see, and doing some bacterial genetics. So I started doing that probably sophomore year in undergrad. And then I continued doing that all the way through graduation. And I ended up doing some honors thesis research with Dr. Zonka there. That was my research mentor. And he really helped me realize that I was really interested in academic research science. So from there, I eventually graduated and decided that I would go to graduate school. So that was a whole another challenge. As I had mentioned, I was the first person to go to even a primary college in my family. So this is something I really didn't know anything about and didn't have a lot of experience with. So going to graduate school was interesting, as many of you are experiencing right now. I quickly became obsessed with genomics. I got into one of the research labs at the Jackson Laboratory in Farmington, Connecticut. And that building was actually being built while I was in graduate school. So this was, I guess, 2013, 2014 era. So the actual research labs were in a different building. So I sort of just walked across the hall from a different lab that I was in and talked to one of the research scientists working there and just kind of said, can I come work with you in your lab? And they said, yes, of course. So I got started working in genomics and quickly, started publishing papers, getting some pretty good data going on. And I was fortunate enough to collect enough data that I could make a poster and apply for some funding to go to my first conference. And that conference was actually the Emaly Piecuch course that's offered up here in Bar Harbor at the Jackson Laboratory. So I was super excited to go and present my work at the Bar Harbor Club. And I was up here for 2 weeks, so I got to really experience the space. And I learned pretty quickly that I was interested in being able to get here. So in my mind at this time, I was planning on coming up here to become a postdoctoral researcher or perhaps some other sort of lab, associated technician. So, time passed. Graduate school was hard. I enjoyed doing lots and lots of research, but I also realized I was interested in informal education and giving talks. And I got some opportunities to teach 8th grade students and some other sort of, atypical sort of teaching opportunities. And I realized that was where I was getting a lot of compliments and I also was getting a lot of enjoyment. So when it came time to graduate, I started looking for jobs in what's called science education. So I'm going to spend a little bit of time now talking to you about the role that I'm in, and then a little bit more broadly about the sort of field of science education, because it's something that I seriously had very little understanding it even existed until I was in this space. So hopefully it'll be helpful for you if it's something that you might be thinking about yourself. And if it isn't, it's just kind of interesting to know that the government is funding science education projects like that. I think it's a little encouraging to me. Okay, so let's move on. So genomic education, that's a weird word. What does it mean? Why do I care about it? And what do I actually do with my time? I'll answer all these things now. So the Jackson Laboratory, if you are not familiar, I think most of you are at this point, it's a nonprofit biomedical research institution. They have a mission, but the whole interesting thing to me is that it's like being at an academic research institution, but it's also got some different sort of features. So up on the top there, you can see the Bar Harbor campus, and below is the Farmington campus. There's also a campus in Sacramento, California, and I believe expanding worldwide at this point as well. But I did just want to take a moment and let you know about the lab itself. Okay, so within the lab, I work in the education department, and it's actually a pretty large group. Education at Jack spans a pretty wide audience. There's things such as courses and workshops, like the Emaly Piecuch course I was just mentioning that I came to as a graduate student, that are put on, hands-on mouse techniques workshops, stuff like that. Clinical education, they focus more on working directly with doctors and people that are working directly with patients. So they basically are trying to educate that audience. And then there's students and teachers, and that's where my department is. That's where I focus my time, is on students and teachers. And this is from the age range of about second grade all the way up to the undergraduate audience. And I'll get into a little bit more detail of the projects we work on with those groups, but that's kind of where we lie. And then just briefly, I'll mention mini courses are sort of the online digital form of some of our curriculum. And I have worked on some content development for those. And then something you might be more familiar with, the post-bac PhD and postdocs program. So you are probably familiar working with people like Sarah Wojcicki and Meredith Thiemann, and those are sort of the category of education that works directly with PhD students and those. And then finally, we've got like DEI initiatives, which are also near and dear to my heart. Okay, so why do I care about genomic education? Well, let me tell you. For over 20 years, DNA sequencing has targeted the analysis of a very small subset of genes or loci. And that's good, but also limiting. But what we've learned from that is that there is extensive variation. In other words, there is way more variability within genes than we had expected. And a lot of this is DNA sequence variation that doesn't necessarily have an effect. These can be things known as variants of unknown significance. But basically the big theme that's come out of that is that not all variation is deleterious or negatively impacting biology necessarily. So that's a really exciting, focus of mine. And the other thing I guess I want to mention is that genomics is a disruptive force. It is redefining our understanding of humanity and our place in the world, and it allows us to celebrate human diversity. And I think these are really important features of using genomics as a teaching tool. So not only do we get to pull in the science and the molecular biology, but there's these awesome societal, ethical concepts that we can really get at using these topics. So that's why I'm interested in doing my work. So what do I actually do? Here's a picture of me standing around. But I'm not just standing around, I'm teaching professional development. So this is the bulk of my work, is high school professional development. This is a picture from a few weeks ago where we teach our course in the summer to, well, it's usually around 30, and I'll show you a picture of them in a minute, but basically molecular biology techniques. So this is a PCR machine, mini PCR machine, and these are some of the trunks that we pack up and send to teachers. I'll tell you more about those shortly. So besides professional development, I do a lot of lab prep and quality control, which I know doesn't sound exciting, but it does keep me in the lab, and I still feel kind of connected to being a scientist that way. So I think of that as an advantage. I get to work on the data analysis of our implementation, which I enjoy because obviously as a graduate student, you spend a lot of time learning how to interpret and display data, and I definitely enjoyed that. So it was important to me to be able to find a role where I could keep doing, working with data sets. Another part of my job is curriculum development and delivery. I serve as a subject matter expert on some of those mini courses, and I also teach quite a bit of curriculum that I'll tell you about in a little bit as well. We develop online and virtual learning. This was really motivated by the pandemic situation. We wanted to get as much content for teachers in their hands as quickly as possible. So there was a lot of push to develop some of the things we had in our minds pretty quickly over the last couple of years. So I can share some of those successes with you as well. Similar to that are career chats, something kind of like this, where we interview different people at the lab and get them to explain the work that they do, and basically just let students know about the breadth of jobs available. And then finally, the most fun, but probably least significant part of my role is second grade bacteriology, which I have a picture of, but the basis is they're second graders and we're teaching them about molecular biology. So it's hilarious. And then adult learning. So I did teach Genetics 1 last year to adult learners, which was really, more of a lecture-based sort of style thing. So you can see the wide variety of distinct sort of things I actually do in my job. And finally, I lead education interest group because I am interested in education. I think it's really a great sort of other avenue to develop people's interests and also get them to explain, new or interesting topics. So there's like the bullets. Let's go into some more details here. So teaching the genome generation is the professional teacher, high school professional development teacher program that we run. So this is a pretty interesting program. It's been going on long before I joined the department. So over 10 years now, the TTGG for short program has been funded by what is called a SEPA grant. And this is something that I was unfamiliar with until becoming a genomics educator. And this is really interesting to me coming from the academic research world because it's grant-based. So we are applying directly to NIH for money, just the same way that you would for academic research science, but instead the focus is all about the science partnership awards. So, just like how the NIH puts out specific goals for their funding, they also do the same thing. SEPA basically is looking for high impact projects that are going to either increase knowledge or basically confidence in science education. So if you go onto their website, it's nihcpa.org, and I can pop it in the chat after. I just can't do it right now because I'm on the monitor. But basically, you can look through a list of some of these projects, and what's awesome is that they filter them by state. So if you are looking for jobs in a specific area, you can look and find out what SEPA projects are funded, and you can basically contact people directly that way. So TTGG's overall program goal is to increase confidence, attitudes, and knowledge in genetics and genomics topics. So it's pretty broad. We don't have like a really hard set, curriculum goal. It's really just more about exposure and attitude. That's what we're focused on. And the audience, as I mentioned, is high school teachers and students, and the regional focus is rural and urban schools in New England area. Okay, so now I can show you a picture of us from a couple of weeks ago. This is our 2022 cohort of teachers. I'd say there's me, 1, 2, 3, 4, 5, 6, 7 of my colleagues, and the rest are all high school teachers. And yeah, they come from all over New England area. So majority are coming from Connecticut and Maine, but we have some teachers here coming from New Hampshire, Vermont, Rhode Island, that type of thing as well. Okay, and here's a picture of them learning some pipetting skills in the lab. So basically they go through a crash course of all of our curriculum within the week and get basically all the tools and materials that they need to take it to their home institutions and implement it with their students. Okay, so let me tell you a little bit more about the program. Here's the kits. When you open them up, inside you can see there's basically molecular biology stuff. Everything that you need to spit into a tube and collect your own DNA and go all the way through PCR, DNA sequence analysis is included in this box. So it's pretty comprehensive, and some teachers choose to implement certain portions and some teachers choose to do the whole thing. But typically we'll rent these kits out for 2 weeks to a month, and they will, go through it in the classroom. We get a lot of phone calls and like late night emails with images. Can you help me interpret this? So, we're on call during the school year supporting these programs. So I already mentioned this a little bit, but making genetics and genomics accessible and energizing students and teachers by linking the content to personalized life. So that's one of the reasons we're so interested in using the students, letting the students use their own DNA. We do anonymize it after the extraction so that they're not directly sort of analyzing and genotyping themselves. But we find that if we use purified DNA extracts, there's a little bit less of that connection to their everyday sort of life. So the assays we use are all sort of focused on things that might be interest to the students. And the whole goal is really just to celebrate the infinite variation within humans. So we're not trying to do any diagnostics here. We're not intending to, make any broad conclusions. We're just sort of showing, visually the infinite variations within the genome. Okay, so let me just break down the program a little bit more for you in terms of its components. So the lab exercises is what I've basically been showing you that come in the kits, and that's where they learn how to do everything from micropipetting, doing DNA extraction, basically all your molecular biology stuff up until PCR. And then the students are actually able to prepare samples for sequencing and send them to the Jackson laboratory, where we'll sequence the samples and then send them back the Sanger sequencing results. And then they can analyze them themselves. So this is my favorite part, obviously. I've made a set of tutorial videos walking through the actual bioinformatic analysis. So we've had pretty good uptake because of that, I think. It breaks it down and makes it a little bit less of a barrier to entry, because you're using some new software and learning how to look at graphs that you've never read before. But I think the bioinformatics math skills is probably where our program really is distinct from others. And then also, very importantly, we have a bioethics component. So we actually partner with PGED. It's called PG Ed. They're a program run out of Harvard, and they basically develop bioethics curriculum, and they've developed some stuff around all of our assays. So it's everything from talking about genetics of, in history, like eugenics, things like that, to more modern things like genome editing and the ethics of privacy, things like that. So it's really comprehensive. They have a lot of lessons. So you're able to, as a teacher, integrate a lot of different conversations that the students might be interested in. So I did just want to mention here that this is the part where I do get to do some molecular biology myself. So in preparation for the professional development seminars, and in order to send this stuff out to the students, I have to make sure we're sending out high quality samples and that things work. So here's an example of me testing the spit kits to see if the ones that were expired would still work for DNA extraction. The answer is yes. So yeah, I don't get to answer the most exciting scientific novel questions, but it's still very useful to have my molecular biology background, and I do get to geek out a little bit at the bench. So now I just want to tell you a little bit about the participation in the course. So here's a graph that I made. Yes! So I still get to make graphs and work with these data sets. So what we're looking at here is actually the student participation, and I've broken it down based on course. So each different color is sort of a different high school class. So you've got your regular biology, honors biology, AP elective, and biotech. On the left side, this is actually total student counts, and along the bottom is the year. We started collecting data for this program in 2015, and this is recent as of 2022, our final implementation this past fall. You'll obviously notice the COVID dip, but what I do want to highlight is the overall numbers here. So since 2015, the program has trained 211 teachers and has been implemented in 179 different schools across New England, and introduced 17,563 students to modern genetics and genomics. And I think those are pretty powerful statistics to show you the power of the program. Okay, so I don't just do TGGG, even though it is the bulk of my focus. There are other things going on at Jackson Lab, Genomic Education land. And one of those things is development of courses and content. I had just touched on the micro courses a little bit, but I will show you a couple other things that we've worked on in the past, I guess, couple of years. So the COVID pandemic, basically it made us want to make some educational material about how the test works. So we got a lot of questions early on about, what's actually going on molecular biology wise, et cetera, et cetera. So we just ended up making a case study, and we submitted it to the National Center for Case Study Teaching and Science, and it got approved. And you can go check it out, download it yourself if you want, but it was a really cool thing to do when we were in lockdown. We couldn't go into the lab, we couldn't send out any of those kits. So we were really happy that, we were motivated enough and, interested enough to get this done. Coming up next week, we'll be having the next iteration of the Cancer Genomics course. This is a course that's been designed for undergraduate students because there is a gap that was identified in undergraduate education on these types of topics, and not just at a specific institution, but broadly across the country. So cancer genomics is a topic I'm very passionate about. But what we try to do here is go through a sort of case study example with the students and have them put their feet in the shoes of a clinical genomicist. So they get experience with everything from the biology of cancer all the way through the sort of diagnostics and clinical trials associated with current treatments. So looking forward to the next iteration of that. I'll be co-teaching it with my fellow genomics educator, Erica Drace, next week. All right, and then another focus is numeracy. So these are basic math skills. In other words, things like fractions, probability, simple multiplication, and using basic equations. And it's been identified that there's a general lack of confidence in these types of things in all students. So one focus of ours is to try to bring these concepts that they've learned back into a more interesting, hopefully interesting focus, and maybe a more relevant example to their life. So in this example, I'm developing some new content exploring taste perception. So basically being able to taste bitter things or sweet things more, and how the genomics of that works. There's actually some fun math to do there. Okay, I did want to mention the online and virtual engagement was motivated a lot by the pandemic, but we were already sort of thinking about going this direction with a lot of our resources because, as I've highlighted, the TTGG program is limited in its physical sort of proximity. New England is sort of the limit for our network. And, as someone from the Midwest, I always wanted to be able to share our content and programs with, my colleagues back there. So I'm passionate about the online and virtual engagement activities. So I wanted to share a few of these with you. The open house is something that the Jackson Lab has done for a long time, but we converted it into a virtual version. And basically now you can go online and check out lab tours and skill building and watch interviews with researchers. And that was really, it was hard to do, but it was very, very fun. And now all those resources are there, and you can check them out if you're interested. We also built out a genetics learning resources page, which includes all of the assets that I've described to you on the last page, like the COVID case study and all of the protocols that go along with the Teaching the Genome Generation program. But additionally, there's some sort of recommended resources from us. So this was also motivated by the pandemic, people asking us, where is this? Where can I find this? So we just popped it all up on there so people could directly access it. National DNA Day is something that we participate in. My colleague, Christina Valianatos, is deeply passionate about this, day, but it's supposed to just be a global celebration, but really we focus on the NIH's sort of goals. But in our group, we do a essay contest, and we usually have a couple other fun activities going on just to do some engagement with the public during DNA Day. Lab Exchange I'll mention a little bit more shortly, but we've recently started collaborating with their platform. They're out of Harvard, and they basically host all sorts of interesting interactive simulations and movies on their site. So we recently were able to get our content up on there for students to find us. We do career chats, like I mentioned, something kind of similar to this. And then finally, this is one of the micro lessons that I've been working on. This is, again, on the cancer genomics topic, I told you. But there are basically a lot of different things going on online and virtual engagement-wise these days for obvious reasons. Okay. This is sort of the final category of the things that I do in my work. Maybe my favorite, I don't know. It's always fun to work with people outside of the institution and also get the perspectives of other people's goals. So I do enjoy external collaboration. This one's more outreach. There's the second graders learning about bacteria. So we basically will go into their classroom for 2 or 3 days and discuss with them where germs might be found, and then do some experiments where they wipe their hands first, then wash them and wipe them again, and then compare and contrast the growth on those Petri dishes. But yeah, don't open those. There's some icky stuff, second graders. Mighty Mice in Space and the Challenger Space Center was an exciting external collaboration. So a JAX research scientist, Se-Jin Lee, actually collaborated with NASA to get some mice up on the International Space Station a while back. And part of that was developing some educational content around it, and then expanding some of the data of the experiment to the Challenger Space Center, which is actually a spot here in Maine where they have, I think it's usually 5th grade age kind of students come and simulate space missions. So they wanted to use some of the Mighty Mice in Space topics. So we got to work with them to develop that content. Here's the better picture of lab exchange I was mentioning. So you can kind of see the idea of the virtual simulation. So what you're able to do with your mouse is actually pick up that pipette, move it around, pick up a tip, pick up solution, move it into the well, run the gel. So it's a fully interactive simulation, not only for gel electrophoresis, which is what's shown here, but we're working on doing this for all of our protocols. So the DNA extraction and everything as well. So that's really tedious, but really exciting, because then you could be anywhere in the world and get this content. I did want to mention the Upward Bound group. We work with them to basically support their program. And then Educate Maine. They're highly motivated to keep Maine's future workforce here. And we work with them a little bit to, well, basically just deliver some content. Okay, that's the end of my talk, but I do have some job hunting tips and tricks for you. And I also need to talk about mushrooms. I can't believe I got through 16 slides without talking about mushrooms. So here we go. Job hunting is kind of like mushroom hunting, and I'm going to tell you 4 reasons why. Timing is everything, because sometimes things just pop up. That's how I found this role. You know, I was looking at museums, I was looking at more public-facing institutions and trying to find roles like that. And then I saw this job was posted, you know, during my job search, and I said, hey, that's everything I've been looking for with a lot of the skills I've already developed. So sometimes it's about just waiting a little bit. I think this is a really good analogy. So looking for certain types of trees first. This is a real mushroom hunting tip, because a lot of them do coexist, or they tend to be in similar locations with certain trees. And it's a lot easier to find a tree than it is to find a mushroom from a distance away. So I think the analogy to that would be, you know, you got to get some help. You got to find the people who can support you when you're on your way. So for me in graduate school, I ingrained myself in the education department. I was always volunteering for those talks and asking what, you know, I could do in terms of outreach during graduate school, because it was just something I was really, you know, I wanted to be close to. And I would definitely agree that that's like a pretty good tip. Okay, you got to know what you're looking for, and you got to keep your eyes open. So things are not always what they seem, and they can be found in unexpected places. So the lesson here is, you know, as I had mentioned, I was looking more at public-facing institutions, and I thought that was what I wanted. The more that I did the interviews for those types of jobs and talked to people working, I realized that, you know, the science was going to become a lot more broad, and I wasn't going to be able to use my knowledge of genomics. And so that wasn't exactly what it seemed like to me. You know, it was unexpected. But yeah, basically, you know, keep your eyes peeled. Okay, and last but not least, there are lots of lookalikes. So try a small bite first, which means take those informational interviews, gain any experience that you can through your other work during graduate school or when you're doing other stuff, and really listen when people talk to you, when they give you compliments or ask you to do things. Is there a certain theme going on? You know, is there something that you're particularly good or interested at? that other people take notice of. Those are I'd say the top 4 mushroom related job hunting tips that I can share with you today. Awesome. Well, that is all that I have in slide format for you, but it looks like there's plenty of time if there's any discussion or questions that we want to have. And I did want to share with you my email as well as my LinkedIn profile here, and encourage you to contact me if you have any questions specifically about any of the work I'm doing as a genomic educator, or if you're looking for any, help or, advice on job searching. So with that, I'm going to end the show, and thank you for your attention.
You have a question in the chat?
Awesome, thank you. I'm just heading over there.
Or do you want me to read it?
Oh yes, I see it.
Okay.
This is a very broad question, but do you think all the educational outreach to encourage people to enter the field of molecular biology and genomics will lead to a tipping point where there will be a surge in new biomedical discoveries sometime in the near future? I like the way you're thinking. I sure hope so. I think more than anything, generally, hi, Dr. Das. Generally, more knowledge about genomics. I think it's because there is just so much more genomic data being collected, and it's becoming more relevant to everyone's lives. I think that's going to be like the most fundamental, reason that I'm interested in encouraging people to enter biology. But man, I really like your vision. I would love that to happen. Yeah, Rachel, great question. So as far as other positions that are similar to the one that I'm in, let me throw the link into the chat about the NIH SEPA grants. So this is what I would encourage you to do. If you're interested in jobs along these lines, check out this website, check out the projects, and what you can actually do is look at the institutions that are being funded. And a lot of these are going to be academic research institutions like the University of Wisconsin. However, a lot of them are going to be small non-for-profits. And then there's medium and large non-for-profits like the Jackson Lab. And then there's also categories of the like the museums that I was discussing, or some of them are just sort of non-for-profit programs that are run, specifically in one city. But that is, I would say, probably the premier resource that is not Indeed, where you can actually go and look at what these jobs are and what these people are doing. There's also conferences like, it's called SciEd and NABT, or I'd say the top 2, non-academic research conferences that are basically made for educators. So SciEd is all of the people who have SEPA grants, they go to that. And then NABT is the National Association of Biology Teachers. So that's where you're going to meet a lot more of the, the professors and stuff like that. But SciEd, I'd say, check out some of the abstracts, maybe from the recent meeting, and you can get a glimpse at some of the projects that are going on that are grant funded like this. Yeah, that's a really awesome question. Thank you. I will also give you the link to the genetics and learning resources page that I was telling you about, just in case you're interested in learning more about that. Awesome. Well, if anybody has any other questions, please feel free to throw them in the chat. But like I said, I'll always be available virtually. You can email me, and I'm happy to discuss anything and help you if I can.
Thank you so much, Emily. You did a great job. I hope everyone really enjoyed today's seminar and learning about Emily's career path and some career paths that may be of interest to you. So unless there's any other additional questions, I think we can conclude today's seminar. And you can have 20 minutes back.
That sounds like the right thing to do on Friday, Brittany. Thank you so much for attending, everybody. And thank you so much to the organizers again for the invitation.
Thank
a
you so
great
much
day.
for joining us. Yeah,
Mm.
it was a great talk.
Thank you, everyone.
Okay.